Welcome friends. Today again we are going to talk about the continuation of series of embryology. Now we will talk about the week 2. We have already talked about the week 1. You can see in the previous video. Now we are going to talk about week 2. So week 2 process will start from the implantation. In previous one we have made the blastocyst formation has been already done and it has moved towards the uterus. So now the implantation is going to be occurred. So implantation is the developing embryo, that is blastocyst, contracts the uterine wall and implants there until birth, facilitated by the internal changes in the endometrium of the maternal uterus. So you can see a diagram. There is a endometrium, endometrial blood vessels are there, endometrial glands are there, endometrial epithelium is there, and slowly the blastocyst has moved from fallopian tube towards this side and it has got attached. ये चिपक गया है जाके on the wall of endometrium in the stage known as blastocyst. A condition is there, a clinical point, that is ectopic pregnancy, a high-risk maternal medical condition that occurs if blastocyst implants in a location outside the body of uterus, such as ovary, cervix, abdomen, or fallopian tube. The latter is most common and is referred to as the tubal pregnancy. मतलब अपनी जगह से अलावा in the uterus wall, if anywhere else the implantation हो गया, then what will happen? It will cause the ectopic pregnancy, and fallopian tube ectopic pregnancy is the most common, or normally it is called as tubal pregnancy. Next, what will happen? We'll see an animation of implantation. You can see slowly the video. You can see first of all it moves towards the endometrium. There are blood vessels which are engorged with blood, endometrial epithelium, then embryoblast cells, trophoblast, blastocyst cavity. It is already present in this particular stage. Then it will go inside the embryoblast slowly with the help of formation of syncytotrophoblast, and there will be cytotrophoblast. Then it will move inside more. At same time, there will be a two cavity will be formed, amniotic cavity will be formed in between the embryoblast cells, and the primary yolk sac will be formed in the lower part. Then slowly it will be completely inside, and a stage known as bilaminar disc will be formed. Then second yolk sac will be formed, and extra embryonic mesoderm will be developed as the implantation is occurring. Then you can see some cavity-like coelomic spaces are also developing inside the embryo. Amniotic cavity is developed. Then there will be a series of changes where some primary villi will develop, chorionic cavity will develop, and it will be completely implanted inside the mother. So implantation is completed. Then there will be one more process will happen. A connecting stalk will be formed, which will be for the future development. And this process will be complete when at the point of entry, a fibrin collagen will be also formed. This is not shown in this animation. A fibrin collagen will be also formed, which will be repair the wall. Now, the implant is fully inside the endometrium. Always remember that while the implantation is occurring, the two-stage layer, that is bilaminar formation, is also happening together in this particular cell or stage also. So many stages are going simultaneously, but we can view only one by one. So here it is for the implantation animation. Let us see again this. You should keep watch on what is happening, what are the names of the cells, what is labeling is here. Syncytotrophoblast means a certain number of cells which are not differentiated by cell membranes or cell walls. So only nucleuses can be seen. So wherever you see the term syncio, it means without cell membranes. It helps in going inside, invading the mother cell or mother epithelium. As these things are happening, you can see in the diagram that other processes are also going on. That is formation of bilaminar layer, then the formation of gastrulation. The next process is already started when the implantation process is going on. So this particular thing happens. Next, we can see the implantation stages. So now we are going to talk about the implantation stages, which is going to start from day 6 or after 1 week. So this we have already seen in the previous slide. The blastocyst attaches to the endometrial epithelium after the degradation of the zona pellucida. You can see the diagram. The blastocyst is also called as conceptus, and the actual size will be 0.1 mm. As the implantation is going to occur, the embryonic pole is also decided. Then on the day 7 or next day, what is going to happen? The trophoblast begins to differentiate into two layers. The trophoblast, the outer layer, it will form the two layers. Cytotrophoblast, which will be inner cell, mononucleated cell, and syncytotrophoblast, which is outer multinucleated zone without distinct cell boundaries. What I have told you previously. Syncytotrophoblast will implant in endometrium. So first of all, syncytotrophoblast अंदर जाके adhere करेगा. Then it will pull other parts of the blastocyst inside the endometrium. Then next day what will happen? Syncytoblast will implant deeper into the endometrium. Embryoblast now will differentiate into epiblast and hypoblast. You can see this particular part. Now two layered part will be forming a disc-like thing between two cavities. Now what are two cavities? Amniotic cavity inside the inner cell mass and the yolk sac, which has been converted from the blastocyst cavity. And this particular disc-like arrangement is called bilaminar embryonic disc. The upper cells epiblast, lower cells will be hypoblast. It will be called as bilaminar disc. Then what will happen? Next day, the extracoelomic or extraembryonic mesoderm will be formed. Extraembryonic is which will be other than the embryoblast cells. And number 10 will be from the day 10, embryo is fully embedded in the endometrium. That's why when you have to take when the implantation is going to be complete, it is day 10. And when the implantation is going to start, it is basically 6th or 7th day. So you can see here a bilaminar disc is formed. Then this primary yolk sac is converted to secondary yolk sac. These are syncytoblasts, amniotic cavity is present, and this the pink color thing, dark pink color thing is the extra embryonic mesoderm, which is in future is going to be divided into further types of mesoderm, and different parts of the body are going to be formed from this particular mesoderm. Remember, this is extra embryonic mesoderm, and this is bilaminar disc. So bilaminar disc, you can see here in the zoom diagram, start to form at the beginning of third week. Means it is at the end of second week. You can understand. First sign of gastrulation means if three layers are going to form, that is ectoderm, endoderm, and mesoderm. उससे पहले क्या बनेगा? Two layers, epiblast and hypoblast, and will be called as bilaminar embryonic disc. And after this, what will happen? Further division or further development will be also started. So you can see here epiblast will form the embryo, hypoblast will contribute germ cells and early blood production because इसके पास अपना कोई अभी supply नहीं है. Then there is umbilical vesicle will be formed. Then in the part of amniotic side, you can see a groove-like thing is going to be developed slowly, which will be known as primitive streak. And this primitive streak is only going to give the first sign of gastrulation. So this is the about the bilaminar disc. Next stage in the implantation only you can understand that on the day 12, some coelomic spaces will be developed in the extra embryonic part. You can see these small spaces are developed. That is coelomic spaces in the extra embryonic mesoderm, and the chorionic cavity will be on the 14th day will be formed. Then the last part. Now chorionic cavity is fully developed. As you can see, chorionic cavity is fully developed and embryo relate attached to its wall by the connecting stalk. Is this the connecting stalk through which it is attached? Now it is inside a fluid-filled cavity. You can understand. Now there will develop a primary chorionic villi to provide the maximum contact with the maternal blood. These are the primary chorionic villi, and this primary chorionic villi in the future will help in the formation of placenta also. So this was all about the second week development in the embryology. We'll continue for the third week in next video.